Great. So thanks for having me here today. I was doing a little bit of reading into your group and learning that it's not just uh, solely on science, but on multiple different facets of it. And so instead of giving a more scientific driven talk, the way we'll have some of that, I thought it might be more worthwhile to give a different perspective on working in the marine sciences from instead of a university or big nonprofit, from just a tiny little organization that's found its way into working on some pretty cool topics. Uh, in case you didn't know what a PALA Marine Research Group was, that's our tiny little group. We're about 10 years old. We just celebrated our 10th year last month. There's only a few of us. We have two full-time folks, and as of this month, a third part-time employee, which is pretty exciting. Most of our funding comes from state and federal sources, and we don't have any real substantial infrastructure. We don't have a lab or an office or a big research platform or anything. So when we formed in 2016, we were ahead of the game working remotely unless we're in the field. And our three primary work areas are in recovering abalone, monitoring of ESA listed rockfish in the Sailor Sea, and monitoring and restoring open coast eelgrass habitats. And I'm going to go into all three of those today and try and give you some understanding of how we came to be involved and have a role in them over the years. Um, but I'll start with a slide I usually uh, provide to undergraduates in a how to get a job talk. And I just give them some pretty tough news that there is no such thing as a perfect job. Any job that you've kind of fantasized about, whether you're an influencer or a professional athlete or a super handsome actor or a professional video gamer, all of those jobs have major shortcomings that you likely don't know about. Not that I know from experience, but nothing's perfect. Everything's a trade-off. And if you imagine your perfect job on axes of your income and the amount of effort it requires and the satisfaction, if you imagine in my poor three-dimensional rendering that there's this job you can find in the classifieds that's going to give you the best of all those worlds, you're probably going to end up unhappy. So that might seem an unpleasant truth to share, but I actually find it quite freeing because as soon as you appreciate that, you can start to get going on making what you have as much of what you want as you can. Uh, and to use myself as an example here, um, my initial title for this slide was "Your plan's a joke, but you should try anyway," and I felt like that was too aggressive, so I softened it to "Plan A rarely works." My plan started when I was 6 years old, and my parents let me watch Jaws, which made me want to become a marine biologist, and I maintained that desire through college and grad school in Miami, working in the Everglades, which was unpleasant to say the least. And then in 2009, I followed a girl out to Southern California and began working in as a contractor on habitat policy at NOAA Fisheries. And this is a point in my career I felt like I had kind of made it. I was kind of where I wanted to be. Um, NOAA was going to be a stable employer. I was going to be able to stay there for a long time and work on conservation related issues that meant a lot to me. And I would get to call myself a marine biologist, which, if nothing else, would allow any number of people who I've just met to ask me if I'd ever seen that Seinfeld episode, and that's what this is really all about. So, uh, but unfortunately, that didn't work out. In that position did not turn out to be a long-term plan for me, and I began working at Power Marine Research Group in 2017. And don't worry, I am not going to gloss over the juicy parts of it. The reason that the job at NOAA didn't work out is that when a federal position is created, if anyone hasn't gone through a hiring process at USA Jobs, you fill out your online expertise. In this case, the job was created for me. It was my exact CV, but it was also a job that was allowed for veterans' preferences. And so the only people who were allowed to have an interview ended up being those who had been serving in the military, which I have not done. And so for the job created for me, I wasn't even able to get an interview, which was quite uplifting. And so I had a choice. I could just relish in the sweet, sweet victimhood and just uh, tell people my sob story. And certainly there was some of that, but there's also uh, the realization that there, as America's dad, Kyle Chandler would put it, um, the federal government still needs help. The work still needs to get done. Uh, there's a lot of it that needs to be done. And so instead of wallowing, I had an opportunity to join with my friend and former business partner, Amanda, working at PMRG on a lot of the areas that I had already been involved in. So back in 2017, I started our super serious two-person team. This is our first offshore survey out at San Nicolas Island, looking for abalone. And I just for those who haven't worked in a tiny company or business before, um, I think there's a lot of misconceptions about it. I think that often people maybe fantasize about parts of it and don't see the full picture. Um, but the best part is that you have freedom. You don't have many people above you telling you what you have to do or don't have to do. We have fewer meetings, and you can choose the work that you want to, and that's highly valuable. And so a lot of folks, whether in academia or government agencies, have expressed that, but they don't always see the other side of it. The other side, we don't have much infrastructure, which can be good. We don't have a lot of overhead to charge on our grants and contracts, but we have less to fall back on. We have no major affiliation. When we apply for grants, no one's going to say, oh, it's UCLA. Well, we know who they are. No one knows who PMRG is. And we are entirely on soft money. So we do need to be constantly renewing the work that we're doing. And as millennials, we went into this not with a goal of making a lot of money. We went into it with the goal of working on goals and restoration of habitats and species that mean a lot to us. And so those are often less lucrative. And so that puts us immediately in a bind for survival. And that's just the bad. I didn't even get to the really bad stuff, which is when you go to school for becoming a biologist, you don't go to school for how to manage a company. You don't go to school for how to manage employees. And so that's all a surprise. You've got to learn it on the job. Getting insurance for workers' compensation for diving and explaining to people that you're not working on an oil rig in 400 feet is not easy. And you have to take your time out of the day to do that because you're super small. You feel like you have to constantly be pushing your work and pushing your results. So that leads to a lot of sacrifice on weekends and nights. It takes tolls on relationships. And given the short-term funding cycles, you're always two steps away from kind of not having much left. And that's true in a lot of soft money scenarios. And so far as I can tell, the best answer is just to try and shove down that existential dread as best you can, because it's always going to be there. Fortunately, for those of you that are still on the call, I'm not just going to whine for the next 30 or 40 minutes. I'm going to take you through each of our three areas, abalone, rockfish, and eelgrass. But I'm not going to talk to you like I'm some conquering hero in the same way I don't say I never get seasick. Just so far, we're doing well so far. So please no one misconstrue that I feel like I've got everything solved or I don't have anything left to learn, because that would be completely false. I'll also give a disclaimer that in order to fit in all of the information from these different work streams, they will be abbreviated stories. And so any one of them could be an entire talk. And so if you feel like you're being shortchanged on your favorite species or anything, I apologize. And you can reach out to me separately, and I'd be happy to share with you about more of what we're doing. Start with abalone. You don't need to dig too deep into these slides. They're actually what I use for new employees to show the development of the work. So the real takeaways here are to provide a timeline of how the work started for us at PMRG, whether we were in the company already or uh, we hadn't yet formed it. And the point is that it takes a lot of time to start up the work, even if you're uh, somewhat involved in various agencies or groups. And so Amanda and I were working on abalone recovery by going out on the weekends and doing surveys on these tiny zodiacs off of Point Loma and La Jolla. And in hindsight, it seems uh, not super comfortable or fun. But we gathered a lot of really valuable data. And the more we did that work, the more valuable we became. Amanda did her master's on Pinto abalone at Fullerton, and I started working more while at NOAA on white abalone. And so once we found that additional funding and had success, then it leads into additional work. For anyone who doesn't know the story of abalone in California, it's a very common one. They were loved to death. They're culturally and economically quite important. You can see the stack of shells behind these dapper gentlemen. Uh, these were all harvested. There's 7 species of animals of abalone in Southern California, and they were the victim of what you might call serial depletion, where one species was over harvested, then the next, and it led to the listing of white abalone as the first invertebrate to have the dubious distinction of being listed under the ESA in 2001, and black abalone followed not long behind. A incredibly coarse review of an ESA policy. Once a species is listed under the ESA, the responsible agency, either NOAA or the Fish and Wildlife Service, has a responsibility to bring it back to a sustainable level. And part of that is developing a recovery plan, which provides a blueprint for any action that may be required. And in the case of white abalone, step 1 is just find out what's left and make sure nothing more happens. And along with that, evaluate the habitat that they exist in to see if there's anything characteristic that can be afforded additional protections. And then in the case of white abalone, there's so few of them left. When you find them, they look like the one on the top right. And they're large and they're covered in fouling, and there's usually no other white abalone within a large area, which is very bad for a broadcast spawner. In this case, it means that breeding or reproduction in the wild is highly unlikely. So in the case we do find animals that are all alone, we can bring them into a captive breeding program, generate a whole bunch of juveniles, and then take them back out into the ocean. And that's a lot of what we've been doing, is taking them back out into the ocean. And of course, all along, giving talks, spreading the good word about all the importance of abalone. Step 1 for us was finding out where they are, and we have done heaps of surveys. This is actually a somewhat out-of-date map. It needs to be expanded because we no longer work from San Diego to Santa Barbara. We need to include the offshore banks at Tanner and Cortez, a whole bunch more work at Catalina Island, and then surveys all the way up to Point Conception. And part of the value of this is that the going wisdom for white abalone when we began these outplant efforts was that if you wanted to find them, you had to go way offshore, you had to go super deep, you needed a ROV or a submersible. The shallowest you may ever find a white abalone is 65 feet, but you really need to be started looking around 100 feet, about 100 to 180 feet to find any of them. And our work, starting with some of those Zodiac trips in San Diego, found that that's actually not entirely true. And so we were able to use scuba diving mostly within recreational limits, mostly within 130 feet or so, but sometimes a bit deeper. And we were able to find that there are a number of animals that exist closer to shore. at Palos Verdes and off Point Loma, and also in depths as shallow. Well, it, uh, could be as shallow as 20 feet, but that was an outlier. But 50 feet or so, you could have a good chance of starting to see any. And that is so incredibly valuable for us because if we want to perform recovery actions and return them to the ocean, then we don't have to go all the way out to San Clemente Island or Tanner Bank. We can start doing work much closer to home and be much more judicious in using our limited resources to have success. And we ended up being able to publish that a few years ago. It's not just regional questions about where to go. It's also a question of what habitat is worth putting baby abalone at. This question is further complicated when only a tiny fraction of the animals remain. Even one individual in a dramatically different habitat can drive your data. And so you have to be very careful in what you include and don't include. An example for that, a white abalone was found on the Port of Long Beach breakwater on the inside of it in shallow water. And if one were to not think carefully and exclude that habitat because there's almost no good reason why that animal was there, you would completely change your understanding of healthy white abalone habitat. And so we take a much more broad approach, looking at both physical features and biological. And physical features, they actually like low relief habitats. So all the gorgeous, stunning, high relief pillars with fish all over are actually not ideal for white abalone. So we want our sites to have mostly low relief habitat and primarily bedrock or boulder, but occasionally a little bit of sand can be good because it channels detrital algae, which they like to feed on. And they also, speaking of food, we need to have a habitat that is full of algae. I can't tell you whether one brown algae is better than another, but we keep track of the canopy kelps and the understory algae just to make sure that we're going to put the animals a place that they have a chance to feed. All right. And so after all that work to find out where the animals live, what make a worthwhile outplant site, we settled for our initial outplants on two areas, one site off of Palos Verdes and one site off of San Diego. But you can't just stop there. You can't just go and plop a whole bunch of endangered snails in the ocean. It's highly unlikely to have success given all the additional stressors that they'll be under, not the least of which would be predation. And so we had to come up with a module to put these animals in to shelter them as a sort of a halfway house as they made their way from the Cush lifestyle in the rearing facility out into the unforgiving reef habitat. And so there have been so many iterations of these. At one point, there was actually a bike tire involved. We've learned a lot. Learn by doing, people. And so right now we've set it on two approaches. The first is this modified oyster basket. We call it pod. And you can see it's attached to this concrete base, and that allows us to secure it very tightly because the ocean is often energetic, particularly in these depths. And we crack the one end of it. The end that's facing us here is probably cracked open just a little bit for them to squeeze out. We fill it with kelp for them to feed as they adjust for a few weeks, and then they'll crawl out onto the reef, with the exception of a few that fail to launch, and then we need to kind of encourage them to leave home. So in some cases, snails are like humans. And then we also have used these modified Cal Fish and Wildlife recruitment monitoring devices. They're a lobster trap filled with cobble. We put some PVC tubes in there, and the ends of the tubes dissolve after 24 hours, and the animals can exit into the shelter of their cobble. That little device on the right is a time-lapse camera. It takes an image every 10 or 15 minutes to give us an idea of abalone emigration out onto the reef and also predator visitation. And it's still more complicated yet. It's not just putting your animals in the module and leaving. There's a lot of organization and coordination that's required, and time out on your outplant sites to make sure all of your equipment is where it needs to be, and that it's mapped appropriately. And so there's a lot of preparation. We've got to put our oceanographic sensors out there, our mini dots that take temperature and oxygen data, and then we add our animals, and then we monitor the heck out of it. We've stepped back a little bit from our monitoring schedule because it has been quite rigorous and gets in the way of other outplantings. So we've had to compromise a little bit as we've become more accustomed to the work. And the question everyone asks, does it work? That's a fantastic question. And I don't have a great answer for you, but we can kind of get around the edges a little bit. We're getting better. Our best understanding of mortality right now are empty shells. If there is no snail inside the snail did not make it. But that is just the base level of mortality because shells can get crushed inside a predator. They can get carried away by currents or waves. They can be hidden and not found by us. So it's the lowest level of mortality, and so we've been using this cumulative proportion of shells, so the number of shells divided by the number of animals that we've outplanted over time at several of our sites in Point Loma and Palos Verdes. And I think the most important part of this figure is that over time, in my absolutely brutal color scheme, over time, we are having less known mortality. And I think that's function of a number of things, not the least of which is that we're getting better at this work. And so there are a whole bunch of animals that are unaccounted for out there. And this doesn't even include our first outplants with red abalone when the modules really needed a lot of work, and the number of known mortalities was closer to about 60%. So this is even better than that. So that's awfully promising. And then a bit more tangible. Last summer, we found a site with 14 wild, or well, we don't know, but they were at the time wild, but they may in fact be our outplants. We were waiting for the genetics to come back. But what was so about these animals is that first, they were only about 400 meters from our outplant site, and second is that they were smaller. I've already mentioned that the adult white abalone we find are usually very fouled and large. And these ones had nice clean shells that likely were quite young, and they were also much smaller, indicating they were likely young. And so we have not found hardly any uh, animals of this size, and to find them also close to each other, so close to an outplant site, is at this point anecdotal, but gosh darn compelling, if you ask me. And it also might help us understand what happens to these animals when they are outplanted. They may not stay immediately on the site, and so that will force us to adapt our monitoring to better track success over time. When you're working with a heavily depleted endangered species, you are in quite a data poor environment. So you're doing a lot of learning by doing and adapting. And for why anyone would want to work with this on this, we have that freedom. We don't have as much time in the office, so we're able to be in the field a lot, a lot. And that also leads to greater technical capability and certification. So it's not just that we do the scientific diving near shore, but we also are decompression trained to do the deeper surveys in 140, 150 feet offshore, which is really important for tracking some of the last remaining populations of these guys. We also are put on the ESA permits, so we can either do the stocking or we can take genetic clips. Here's one of our colleagues taking a swab of an abalone pour out at Tanner Bank, which is covered under the ESA permit. We've done a lot of the science related to the white abalone recovery, whether it's the surveys I've already mentioned or looking at the predator aggregation on the time-lapse cameras, or evaluating how the modules themselves are working. So even though we've only been around for a short time, what we've done in the years has allowed us to stick around more prominently, as my old professor told me. Okay, so leaving abalone and heading up the coast to the Salish Sea, where we work on rockfish. And for anyone who's not worked on rockfish, doesn't really know who they are, uh, rockfish are incredibly cool. First of all, they're venomous, which ups the cool factor of any species. And they're mid-level predators. They live a really long time. They have kind of sporadic recruitment and reproductive success. And this is what a reef might have looked like historically, more commonly in the Salish Sea, but certainly doesn't now, because the story of rockfish in the Salish Sea is not unlike that of abalone in Southern California. Overfishing has led to large-scale population reductions, and that, in the case of two species, yellow eye and Boccaccio, has ended them up on the endangered species list. Boccaccio are endangered and yellow eye are threatened. And just like the white abalone, a recovery plan, the blueprint for all the actions required to bring these species back to a sustainable level, was put together for rockfish. And that's some of those actions are where we came in, because so little is known about the reproductive success and recruitment of rockfish in the Salish Sea. A huge amount of data were required, and we began working on a project to uh, collect a substantial amount of data and integrate with a whole bunch of partners to build out that data set. And I found myself in 2015, 2014, when my then wife moved to Washington for 2 years for a fellowship. I found myself wanting to spend more time up there. I got in touch with the recovery coordinator for rockfish in the area, and as I was a NOAA diver, he suggested that we work on this, and so we started this project from nothing. And it's been quite successful. But you'll note on the bottom, I have the terms life raft on there. That's because this is the one project we have that's been a casualty of our new I'll call it priorities of our government. And so we finished our NOAA grant last year and have been kind of scrapping for whatever funding we can to keep the work going. We've had a little bit of success about that, which is heartening. But it took us 5 years from starting this work to get any kind of funding at PMRG, and then even more time beyond that to acquire additional projects to work on. And as I've already mentioned, it's a big ocean, and it's doing different things all the time. And fish are no exception. Fish are notoriously noisy data. And so the more variability you have in your data, the more data you need to say something. It's a highly simplified conceptual graph, but it largely holds true. So the Salish Sea is huge. For anyone who has experience there, you know that South Sound looks nothing like the San Juans, and add in any number of other regions they're in, and you can say the same thing. And sometimes we get these boom events of rockfish recruitment. We don't know why. And so we've gone out and built a survey team to survey as much as we can. And for those of you that came for kelp, there's a kelp photo. I feel like maybe the most important takeaway from this project is anyone that says just do a citizen science project that will solve your data woes. Please don't do that. It's a huge undertaking to try and involve partners, whether they're citizen or professional, in your survey. And we started this project in 2014. And that entire first year, we were just trying to figure out how to collect the data, and that involved meetings with anyone that took data underwater in the region, whether it was government agencies or academic groups, nonprofits, whoever we could talk to. And we didn't start collecting data until 2015, and that took us a while. You imagine a person from Southern California strutting into the Pacific Northwest and saying, "Hey, would you please take these data? They're important." But we got buy-ins from some amazing partners early on, and with them, we got our 100th survey transect completed in 2 years. And for those of you aware of survey effort and noisy data, that's not going to get it done. You need a lot more effort than that. So very, very bad if that was how it was going to continue, but we strove to continue building our partnerships. PMRG got the funding in 2018. We added even more partners in, and we got our 1,000th transect in 2020. So it took us 2 years to get to 100 transects and about three to get to 1,000. So already we're building a little bit of momentum. We see our first reproductive boom event in the winter of 2021. We start seeing more and more of the ESA listed yellow eye starting in 2022, and that's continued to present. Around that time, Reef Check moved into Washington to start their surveys, and one might think that would be bad for competition, but in this case, it's the exact opposite. We've worked incredibly well with them to build a collaborative survey team, which just makes me so darn happy. We also started working with a group up in Howe Sound in Canada to get a more comprehensive data set from the entire Salish Sea, and that's been rather productive as well. And then in 2023, we also put together a mathematical model to take all of these disparate opportunistic data and distill them into a single recruitment index. And we're working on applying that to all of our now 11 years of data to figure out some of these initial questions. What are the recruitment drivers? And that's incredibly exciting, almost as exciting as getting to our 10,000th transect in 2024. So for those of you that are looking to build a citizen science project, this is about 10 or 11 years of just pushing the rock up the mountain, and then you start getting going, and along the way you get different funding regimes, and these are all points to keep in mind before you start. The effort is I think equally impressive spatially. If you break our surveys into 3 years, the first 4 years or 3-year bins, the first 4 years of the program, we're working primarily in Central Sound up in the San Juans. And that it's fine, I guess, but not enough to get at the questions we want. And so starting around COVID, people decided to get out of their house and survey for rockfish a little bit more. We're building up a little more success, and then the last 3 years, the effort is really expanding, both in the amount of time spent at the typical sites and also expanding out into new areas. You can see Howe Sound, the northern reach of 2022 through 2024, and even some surveys out on the open coast. So that's incredibly heartening. And with all these data we've collected, I wish I could come to you and say that we've solved it, that we have the exact time that the rockfish recruit, and these are the conditions. But it's as you may imagine, a bit more complicated than that. And so we separate these young rockfish into different species categories. And so the first one that are the coppers, quills, and browns, you can find them just about any time of year. They seem to have just a really long range of recruitment, and it doesn't seem to vary that much. Whereas the yellowtail and black rockfish, they definitely have all or nothing events. And the conventional wisdom may have been that you look in the late summer and early fall for recruitment, but not in the case of these guys. The first boom event occurred in the winter and spring, and if we hadn't been surveying all year, we would have completely missed it. And then the Puget Sound rockfish, perhaps the least charismatic fish to exist, they have spike often in the spring and then just disappear for a while. So we got all different kinds of recruitment patterns that occur that were a bit of a surprise for the conventional wisdom. We've also, I already mentioned the positive news about yellow eye starting in 2022. We'd only seen, I think, 2 yellow eye recruits in the existence of the program until then, kind of a freak event in 2016 in Hood Canal. And since then, we've been seeing more and more. And I definitely appreciate that our effort has been going up, so that it's not necessarily as striking as this figure looks. That does not take effort into account. But some of these sites we had surveyed for a long period of time, and we had not seen yellow eye until the last few years. And so it's somewhere between neutral and good news. It's definitely not bad news. And also they are just a very attractive fish, and in a somewhat drab underwater environment as the Salish Sea can be at times, seeing these guys is awfully exciting. So we've been working on that for a really long time, since the inception, and I think the reason why we've had success on that project is probably encapsulated in the one time I've taken criticism in a productive way, in that we applied for a grant to expand our outreach in 2019, 2020 or so, I want to say, and we got rejected. But instead of just getting a form rejection, we received follow up that said, "Hey, we don't know what the result of additional outreach is. If you do 3 talks or guided dives, what does that mean for the long term for your data?" And my initial internal, fortunately, reaction was, well, how could we know that? How could we get that information? No one knows that. And then I got over myself and I realized we actually have these data. I know how much I've been out doing outreach with our partners, whether it's on dives or on presentations. I have logs of all that. And I also know how much people are diving. So uh, we actually can put that together to answer that question. And so that's what we did. And we collaborated with the group that rejected us in a productive way, and put together a publication that featured this plot here. And I think there's a couple interesting things to note from it. Each point is a year, and it's color coded by the organization. And the first point is that no matter how many outreach events you do in a given year, it takes about 2 years to work with a group until they start collecting data in a meaningful fashion. And so that really speaks to building a relationship. And it makes sense, right? If someone walks into your dive club or into your office, your research group, whatever it is, and say, "Hey, will you take rockfish data for me?" You don't know who that person is. You don't know if they're going to stick around. So why would you invest your time in that if you don't know? Uh, and so after a little bit of stability, they tend to get more involved. And then the second, which is not perfectly depicted, but on this plot, but the slopes of the lines increase the later the group uh, joined. And so that means that there's more diving output the later a group joined the program. And that suggests to me either a form of synergy, whether dive groups are talking to dive groups saying, "Hey, this is worth our time," or it's suggesting that we're getting some kind of valuable data, and we can share that with our partners to entice them to participate. All right, so finishing with rockfish and moving into possibly the prettiest area that we work in, which is open coast eelgrass. Um, so I started working at eelgrass when I was at NOAA as a contractor. I did a lot of policy on eelgrass, and policy. There's nothing like working on policy to make you want to stop working on policy and go outside. And so talking with a professor at USC one day, he told me about a eelgrass bed they had at the Wrigley lab, and no one really knew much about it. And so I started working with him and one of his students back in 2013. And I always knew I wanted to go spend more time on those open coast eelgrass beds. I felt like there's probably a lot to learn. And when I started at PMRG, I had that freedom to say, yes, even though we may not be necessarily paid for it, this is something that I think is worth exploring. And so we expanded our work from just Big Fisherman's Cove out to any open coast eelgrass bed that we could find. And that led to a huge amount of data and underpinning some restoration work. And so it's a little bit biased here, but it did take us about 9 years from this initial collaboration to getting any funding. And then from there, this has probably expanded as good as any other work that we've been involved in. And you may not, if you're not familiar with eelgrass, you could be forgiven for not understanding why open coast is any different than what's in the bays and harbors. That's fair. And most of the work on eelgrass in Southern California has occurred in bays and harbors, and that's because there's so much more stress there. The map on the right shows some dredging diagrams of Newport Bay, if you dredge up habitat, you're either going to remove eelgrass directly, you're going to make it too deep for it to grow. And all the docks and piers reduce the light, preventing eelgrass. And so there's only so much area eelgrass can grow in these bays and harbors. And the plot on the left uh, shows that the remaining eelgrass distribution kind of follows away from all these stressors. And even the main policy document of eelgrass, the eelgrass mitigation policy for the state of California, has a list of all the eelgrass transplants that have occurred in Southern California, and they're basically all in these bays and harbors. And I think that we can all join with Jan saying that this is way too much work in the Marcia. Uh, and perhaps there's some other areas that would require some attention. Uh, and Open Coast is definitely fits that. And there are a good amount of them out there. And when you start working in an area that very few people have, you don't need a huge amount of data to make a meaningful impact. Uh, First, I'll start by noting that there's in fact two species of eelgrass that exist on the open coast. There's the Zostera marina, which you'll find in the bays and the harbors, and then there's Zostera pacifica, which is a much more wide-bladed species, and it exists in much higher energy environments. And it can be found quite deeper as well. And so we went out starting in 2018 and just started monitoring through mapping and recording structural metrics like density and length and width, and also recording the fish length and species composition inside these beds. And the first thing we found is that some beds were growing over time. This is Emerald Bay, expanded dramatically. Some were declining. This was at Ripper's Cove on the front side. And then some you could set your watch to and just stay steady throughout. So not all of these, even though the conditions might be largely similar on the front side, these beds are clearly responding to something different. Also, there is a dramatic difference in the fish assemblage that you find across the two species of eelgrass, um, uh, as indicated by this NMDS plot, which for those of you that don't have a background in multivariate statistics, each point here is a survey site, and its location on this plot is driven by the relative abundance of the species surveyed inside that site. And so the further apart they are, the less similar the species assemblages, and the closer they are, the more similar. And so the Zostera marina had a lot more Salima and rockgrass, and those were not the only species driving the difference, but some of the most important ones. And then mid-level predators like barred sand bass and senorita were more common in the the Zostera pacifica beds. But the difference is in habitat function across those two beds weren't limited just to composition. There's also a component of length. Remember, I said it was relative abundance of the species, not entire absence. And so there are a number of species that were found in both two species of eelgrass. So in this plot, we have 5 of the most common fishes that were seen in both of those eelgrasses. And often those that are in the Zostera pacifica, the higher energy, wider bladed habitats, are bigger. In those in the Zostera marina are smaller. And so that suggests that there might be more of a nursery function that exists in the Zostera marina beds, and more of a, say, standard foraging habitat inside the higher energy beds, which is super cool. This entire time, no one knew those two habitats formed a completely different function. But we didn't want to leave it at that. We were collecting all these data, and so we wanted to apply it to some restoration, particularly given that all of the transplant work had been done in bays and harbors. We wanted to try some on the open coast, and so we started with some very low-hanging fruit. There was some existing funding with our partners at the Bay Foundation to conduct some experimental transplants in Santa Monica Bay. And I know what you're thinking, and I was thinking the same thing. What the heck? Why do you think that would ever work? And that's a fair statement. when you know so little about the distribution of these beds, you're forced to start with a little bit of guesswork. And so we chose these sites based on some qualitative characteristics, proximity to a south-facing shoreline or proximity to a submarine canyon that are present in other open coast beds, and we use that to dictate our site selection. And so we transplanted a small amount of eelgrass from massive beds on the backside of Catalina Island into these three different sites. And we had an inkling that this probably wasn't going to work out, that we probably weren't going to end up with huge meadows following this effort. But a valuable lesson in restoration is that, well, even if you have an inkling it may not work, you should still try and take some data that will help you the next time that you do this. And we found that not all the sites, none of the sites succeeded long term, but they didn't all fail the same way. And for example, Malaga Cove, which is the small green triangle, that one did the best. And so we could look using these sensors that we put out both in the restoration sites and the transplant sites. That top rod there, that's a pressure sensor that gives us an idea of the wave energy coming through in case it's too strong for the eelgrass to survive. And then the bottom is a light sensor, an oxygen sensor, and a temperature sensor. And a quick view with our two reference donor beds on the right side and our transplant beds on the left. You could see that, well, realistically, none of them were ideal. And we had the privilege of a red tide in Santa Monica Bay at the same time. You can see that both off of actually all three of those with the dissolved oxygen. Dockweiler, Redondo, and Malaga had a steep drop in dissolved oxygen during our transplant, which was fun for both the eelgrass and diving in a red tide to do surveys. So we were able to take these data and we're able to apply them forward. And so we are conducting a subsequent transplant this summer in Malibu, where we have put these sensors out in 4 other sites and can compare the data we've taken from them to choose sites that are going to be far more likely to have success. And all of our sites in Malibu are way better than these. So we appreciate the eelgrass that couldn't be with us today for their noble sacrifice in promoting further restoration work. Now mute our soundtrack on this, but after our big swing in Santa Monica Bay, we went for a more doable approach in Catalina Island and did a Zoster Marina transplant at a site that we knew had eelgrass until 2016 when a big Santa Ana storm tore it all out. And so this is us in 2022. This is our transplant site. Before we started, we used tapes to delineate planting bins. And so it's mostly unvegetated with a few patches in and around, suggesting that we've got a good chance of success. We worked out of the Wrigley Institute and transported between donor and transplant site. We collected under strict supervision of the locals at Rippers Cove, where we'd monitored for many years and knew that there was a good amount of habitat to donate. When you're collecting eelgrass, one of the most important components is that little stemmy structure right there called the rhizome. That's where a lot of the nutrients are located, whereas the blades are kind of solar panels. We processed all the material into planting units at the Wrigley lab and then transported it back over to the transplant site at Button Shell. We also had some folks that had an ROV that they wanted to test it out for planting. I think they ended up fairly happy, but I'm suspicious of the long-term value of that based on the one trial. Planting eelgrass is just like gardening. You dig the hole, you put the root bundle inside it, and then repeat and repeat and repeat. And when you're finished, it looks just like that. A whole array of freshly transplanted eelgrass bundles. And it turned out that this was about as successful as a project as you could expect. With the naturally occurring eelgrass after transplanting, there was a little over 1,200 square meters of habitat on the site, and we went back and monitored it last month, and there was over 3,400 square meters, so almost a threefold increase following our transplant there, which is super exciting. But it's not just the area that's done well. These are some images of the transplant and the fish using it, and my little rockfish survey heart just fluttered when we saw these young of year Boccaccio, which are not endangered in Southern California, but still very cool to see them using our transplanted eelgrass habitat. And one of the benefits of surveying every other open coast bed we can find at least once a year is you've created a whole bunch of reference sites for your changing fish assemblage. And for the numbers people, we found that after just one full year of transplant, the fish assemblages at our transplant site were indistinguishable from all our other reference areas. And so the first year they were significantly different. You can see that in the top panel that says 2022. Our transplants, our relative abundance of the species in the transplant area were different than all the other beds we surveyed, but after that, they were indistinguishable. So very short turnaround to being a valuable habitat on the front side of Catalina. Why us? I on this, I don't know. I have no idea how everybody else dropped the ball on working on open coast eelgrass beds. There's just so much going for it. It's an incredibly high value habitat. It's got so many ecosystem services. There's seemingly limitless scientific questions that you could ask out there. There's a decent amount of infrastructure, whether you're at the Wrigley Lab or with the Channel Islands Sanctuary or UC Santa Barbara on the Northern Islands. There's a huge policy or mitigation outlet for it, given that the bays and harbors are so overdeveloped, it's hard to find areas to mitigate for any loss. And so if we can get successful transplants going on the open coast, that's a possible outlet for making sure you don't lose any function of that habitat in the region. And it's just often just quite pleasant to be out there. There's giant sea bass, there's giant schools of Salima. It's just a really great place to be. And I tie this up, all these different work streams and my complaining about my job with adapting a statement from my intro to marine biology professor at Miami. It was kind of well known that a whole bunch of kids came in to become marine biologists and very few left. And that led one to believe that it was hyper competitive. And so he told all of us, instead of his look to your left, look to your right, half these people are going to be English majors in 3 months. All he said was, there's always room for the best. And I suppose that's true, but I would adapt that to saying that there's room for those that, like us, refuse to quit and get a real job and just keep going back to the ocean and counting things. And to any universal messages that might be derived from these last 10 years, I think the first is listen to what the universe is giving you. If you have your heart set on a certain job, but that's not working out and there's another opportunity, don't dismiss it immediately. Always be building your partnerships. They're going to become incredibly important, particularly if you're as small as we are. That the work that you're starting out, you might not know where it's leading and it might seem insignificant, but every little bit of data that you take helps. It can lead to large data sets that will be super valuable, whether you're doing habitat restoration or looking at changes in endangered species over time. And I'd be remiss if I did not thank all of our partners, including just a handful I have highlighted here, because we absolutely do not exist without them. Yeah, and that's the talk. Be happy to answer any questions. I don't know what time it is.